Hi everyone, I'm Karen McRae-Johns, a product manager at Jomar Life Research. As a response to COVID-19, many research labs temporarily closed. As restrictions have begun to ease, many researchers are returning to their labs. Today I'll be discussing tips to consider during this time. As researchers are returning, many will begin culturing cells again. My first tip is that this is a perfect time to screen your cells of Mycoplasma contamination. Mycoplasma is the smallest and simplest self-replicating organism. Contamination cannot easily be detected visually or even by optical microscopy. Mycoplasma contamination is problematic for a multitude of reasons. Mycoplasma can drastically alter cultures and skew results, for example, by altering cell metabolism, growth, and even gene expression. Additionally, Mycoplasma can seriously impact data reliability and reproducibility. And as scientists, these are attributes that we strive for when producing data. Mycoplasma contamination is also highly infectious, and this can lead to loss of precious cell lines and loss of money and time. InvivoGen have developed a colorimetric cell-based assay to easily detect Mycoplasma. This assay is called Plasmotest. The assay has a simple readout. For instance, if Mycoplasma is present, you can visually see a color change from a pink color to a purpley blue. This can also be quantified using a spectrophotometer. If no Mycoplasma is present, no color change will be observed, and there'll be no significant change in optical density. In addition to the simple readout of Plasmotest, other benefits include that it detects all Mycoplasma species with a very high sensitivity. The assay can also be performed in a 96-well plate, meaning it can be used as a convenient method of routinely testing multiple cell lines. In addition to checking cell cultures, Plasmotest can also test biological reagents such as media and FBS. Notably, since frozen stocks of the Mycoplasma reporter cells can be made, you will not need to repurchase the Plasmotest kit. In addition to Plasmotest, InvivoGen have a variety of products to detect, prevent, and eliminate microbial contamination, as shown here in the table. My second tip is to effectively eliminate any contamination. For example, if your cells are contaminated with Mycoplasma, Primocin treatment can be used as an effective means to kill all common Mycoplasma strains and safely rescue your precious cell cultures within 2 weeks. My third tip for you today is to use antimicrobials such as Normocin and Primocin to prevent culture contamination from occurring. Normocin and Primocin both serve the same functional purpose of preventing contamination between bacteria, fungi, and Mycoplasma. The main difference between the two is their formulation, which directs the cell types that they are best for. For example, Normocin is considered to be more of a general purpose antimicrobial for cell lines and is not recommended for primary cells. Primocin, on the other hand, is formulated for the added sensitivity of primary cells, which are not as robust or hearty as established cell lines. My fourth tip is to select and maintain cultures with only your desired clones. Adding selective pressure using selective antibiotics upkeeps the cultures and prevents against any genotypic shift. InvivoGen have manufactured sterile and endotoxin-free selective antibiotic solutions, as listed here. And all of these are a convenient way to apply to your cell cultures in a ready-to-use manner. My fifth tip is in regards to routinely disinfecting common sources of contamination, such as incubators, water baths, and surfaces. Promocell have a variety of disinfectants for these purposes. The water required for the humidity in incubators is a common source of microbial contamination. AquaGuard 1 is a solution that is non-volatile and stabilizes and cleans the water. That is extremely effective at maintaining the water quality in CO2 incubator trays. AquaGuard 2 is intended for the maintenance of water quality in general water baths. The Active ingredient in AquaGuard 2 is highly effective to prevent growth of unwanted organisms. This product is stable at high temperatures, for example, 100 degrees Celsius, and doesn't produce foam in circulating water baths. Similar to AquaGuard 1, AquaGuard 2 is non-volatile, non-irritant, and non-corrosive. Additionally, Spore EX Spray is a reliable all-round solution for highly effective surface disinfection of critical workspaces in less than 1 minute. It has been scientifically tested to act against bacteria, viruses, and fungi. It is also shown to be very safe and simple to use. This product is an excellent method to clean and disinfect a variety of surfaces, such as laminar flow hoods, incubators, workbenches, cell storage boxes, and even instruments, making it ideal for everyday lab use in order to maintain safe, clean, and contamination-free work areas. If you have any questions or if you would like pricing for any products that I've mentioned today, please feel free to contact me, Karen McRae-Johns, at info@jlresearch.com.au. If you use the promo code JLR I'm Back with your inquiry, you'll be eligible for up to 25% off until the end of June 2020. Thank you for listening.